value as well. The lower the E value, the better your sequence matches. It's like the probability that this match is happening just by random chance. So yeah, these are a few things about BLAST. Then we also want to know some types of BLAST. So BLAST N, this N stands for nucleotide. So here your query will be nucleotide. Your database will also be a nucleotide. Then we try to find similar DNA or RNA sequence from the database, right? And then we have BLAST P, where your query will be protein and also database will be protein. So this is to find your homologs in your protein sequence or something like that. Then we have BLAST X, which is where the query is nucleotide and the database is protein. So basically your query is translated to a protein sequences, right? And then we have T-BLAST-N, where your query is protein and your database is nucleotide, where your nucleotide database is again translated, just the opposite of this one. Okay, and then we have T-BLAST-X, where both the query and the database are nucleotides, but they are actually translated. So yeah, let's do some practical now. Right. Okay. So I'll just go. The easiest way to find NCBI is just go for NCBI, and you can just Google it. You'll come across this site where you have to go to this ncbi.nlm.nih.gov. So this is the website for NCBI. And here you'll find all the list of resources, right? So all the databases here, and they have a list of other containing databases like nucleotide, protein, or the PubMed. PubMed is basically for the literature. Then SRA for you have SRA for this expression, or GEO for again, we do this transcriptome or microarray analysis. So like this, so you can just get familiarized with this one. This is a beginner's friendly, so we won't go for details here. I'll just show you how to use it. Okay, if you just write insulin, let's say you want to know more about insulin, or you can just type insulin here and it will give you all the information it has regarding insulin. For example, the number of papers that are published, the GEO data, it's the microarray data sets, or your transcriptomic data sets. So you can do some patient versus disease type of comparison, but those we'll cover in upcoming sessions. These are more advanced topics. So let's stick to the easy ones. Let's see what a nucleotide database gives us, right? So when you go to nucleotide, you can also select it like this. By selecting in the database section, you can come to nucleotide and select nucleotide and go for insulin. And then here, this page, when it pops up, you will see on the left side the species name. These are the species where you want to check. Or if you want to specifically go for some other organism like humans, suppose, you can also do it here by going to advanced, and then you can go for Homo sapiens maybe. So this will give you only Homo sapiens. So on the right here, you see this Homo sapien comes automatically under the organism section. So this is called mesh terminology, but again, we'll discuss about it maybe later. So basically, these are the hits that you get for your insulin nucleotides in Homo sapiens. So the first one, as you can see, is the base pair length and then accession key. This accession is actually nothing but your like a roll number. Let's say a primary key. In every database, there is something called a primary key. So primary key is like the roll number. So in a class, no two roll numbers can be same, right? A person's name can be same, but the roll numbers will always differentiate between the person. Same way we use accession codes here. So this accession code actually differs between one sequence to another. So if we want to just go and check in one of the sequence here, let's say we select this. So we'll end up in a GenBank database page, right? And it'll tell us everything that we need to know about this insulin. right, where it is, how long it is, right. And then we will also find the authors who deposited it. Then we'll also find the journal that they have published regarding this sequence. And if you scroll down, you'll see this nucleotide sequence over here, right. So there is one more way to get your nucleotide sequence, as I mentioned, was FASTA. If you click on FASTA, you will get the nucleotide sequence like this over here. So you can just copy and paste this one and to your notepad or a flat file, and then you can start working on it. And the first line of a FASTA actually tells us, this is called a FASTA identifier or a FASTA header, which tells us about the sequence, what this sequence is, right? This is the locus, this is the species and stuff like that. You can put anything over there. This won't be directly considered in your analysis in BLAST or something. It will be kept as an identifier, and the remaining of your sequence will be taken for the analysis. A FASTA header is marked by this sign over here, a greater than symbol. And yeah, that's how it works. If you want to save this file, you can also do a send to here. You can do send to file and then FASTA and create file. So this will create a file that you want. If you want to call it insulin, let's say, and then save it as a FASTA file, you can save it just like this, and your file will be saved as insulin.fasta. Yeah, so that's how you can get your data, get your nucleotide sequence from your from the GenBank or NCBI, right? So let's discuss some more what we can do with this nucleotide sequence, or what we can do with these entries. So let's say we want to go for this gene over here, right? So this will give us where the gene is located in this for insulin, right? So these are the depositions when they were assembled and when they were submitted to NCBI. Right here, you will find some details about it, like the HGNC symbol. This is this is a standard symbol, a symbol for all the genes. So insulin is called by this name. And basically AGNC you will be using more when you are doing into some pathway analysis. Let's say you are working on some drug and you want to see which of the genes are actually affected by the drug, or in a disease condition which of the genes are actually affected. So you have to understand the pathways, and those pathways are actually part of this AGNC symbol, or there are other contrast symbol and stuff like that. Right. Then here we can find other information as well. One of them is the lineage, the hierarchy of the sequence, of course. And then if you scroll down, you will see this location of the gene where it actually where it's present. So the introns and the exons are marked here. And if you scroll further down, you will see something like this, where the expression of that particular gene is shown, like in which of the regions the particular protein is expressed the most, right? So you can work more on this when, according to the question you're solving. For example, you're trying to see a protein's expression in a given scenario, which a tissue is expressing it more during a disease condition. You can refer to this kind of your graph, which will be your starting point, right? So if we scroll further down, you'll see something like interaction here. So this interaction will tell us what that particular gene is interacting with the other genes, right? So what our gene of interest, this insulin's gene, SLC, that gene is actually having interactions. These are the other genes that it is interacting with, right? So yeah, if you have any questions, just paste it in the chat. Maybe I'll take it up when we have time. And then I'll also try to show you something called variation here. I don't see it, but it should be there somewhere. Yeah, variation viewer. So here you'll see the variation of this particular gene, when they have some variations in it, and that causes some problem, right? So for example, if there is some mutation in this gene, then, and it becomes something of, a pathogenic one, then you can see these are the entries for which we know that a mutation in that gene actually causes some disease, right? If there is some single nucleotide variation like SNPs, so one nucleotide actually changes in the sequence, what it can lead to, right? So you see here there are some entries for it. For example, if we go and check this one, um, we don't have a clear picture. Transcript change is not written here. Maybe we'll check this one. Okay, we don't have for this entry as well. But basically, it will tell you, yeah, here we have this entry, right? So you see, this variant allele is C. When this T is changed to C, you will get this disease. So I think we are looking into some a variation of the genes. But anyway, the protein that changes is serine to proline. So in this position of the gene, the amino acid actually changes, and that's why you get this disease. So there are no much information about this variant, but anyway, this is what the SNP does. You can understand how one change in the letter of your DNA sequence can lead to a complete change in the protein and finally lead to some disease. So this can be found in the SNP as well, right? So yeah, we have discussed still variation. Now let's do some BLAST then. Let's go to BLAST and NCBI. Right. So when you open BLAST, you will see this landing page. As we discussed, there these are the types of BLAST, right? Nucleotide, protein, BLAST X, T BLAST, and T BLAST X as well, but it's not shown on the landing page. But anyway, let's go to the nucleotide BLAST. So here you will see T BLAST X. These are the 5 kinds of BLAST that you can do. We'll just stick to BLAST and for this tutorial. So here you paste the nucleotide sequence that you have, right? So we just downloaded the insulin structure, right? We'll go to the insulin.fasta. Let's see if it takes it in, else we'll just copy and paste it maybe. Right, so here you can also mention from which region of the sequence you want to check, right? Then you can also give a title to your search, let's say insulin testing, something like that. And then you can also check which database you want to go for. This core nucleotide database is the entire database set. Then you can also go for RefSeq, which are more curated versions of it, and so on and so forth. So if we want to go for the nucleotide collection, like the entire nucleotide here, we can go for this one, right? And then we have some more optimizations like Mega BLAST or BLAST, and this is a comparatively newer one, Mega BLAST. When during our time we just had this BLAST N, but anyway, we'll go with the newer one. There are other things as well that you can optimize. For example, number of maximum target sequences, let's keep it to 50 maybe. Then expected threshold 0.5, word size 28, fine. And then we'll just do the BLAST, and we also want to open it in a new window. So while we wait, we'll see, it takes some time, but basically it will try to align your query sequence with the database that we have, right, in the BLAST. We are currently doing with the nucleotide database as a whole. So yeah, it'll take some time, but until it completes, I also wanted to let you know that this BLAST is not strictly for NCBI's landing page. You can also perform BLAST inside your local system. So let me just quickly show you maybe, but not in detail. I'll just show you how it is normally done. So basically, right. So if you're familiar with Ubuntu or Linux, let's say you're familiar with Ubuntu. So here you can install BLAST and do your BLAST locally. Right. So suppose you're working in a lab and you have a database which is not present here in the NCBI's database. You have not submitted it, and you have sequenced some, bacterial colony or something, and these are just proprietary to your lab, and you want to do the BLAST and see if these sequences match. So you will do a local BLAST because this BLAST won't give you any results, right? So there are ways to do it. And the best way is, first it comes in NCBI's BLAST Plus package, which you'll have to install, and then you can work with it. So first you need to make just a second, either blast. Yeah. So these are the two methods by which you can do either a nucleotide blast or a protein blast. So there are a few things that you have to do before doing it. So you have to give a db. We can do this in the next class, but I just want to show you how blast is done. So basically you have to first select this nucleotide blast, let's say blast n, and then you have to make a database. So that database will be your personal database or your curated database that you get inside your lab that you just sequenced or something. You can make this your database and then use the query sequences that you just did. Let's say you did some PCR and you got some sequences, and then you'll use that query sequence in your database sequence and run a blast like this. Currently it's giving me an error because we have not set any local database for it, but maybe we'll show you in the next sessions. Okay, so yeah, we have some results over here that after blast. So here you see the best hits that it got, right? Here is the maximum score, the total score, the query coverage, how much of the data was actually, aligned. Then we have the e value. As I told you, it's the p value, the lesser the better. Then identity, this is 100% because it's the same one that it's the same sequence that we use. And then there is this accession to that particular entry, right? So let's say you want to know something more about it. You can also go for this graphic summary where you will see this is your query and how it is aligned, right? Not everything will be aligned well, but in this case, since we are using a very known protein like insulin, that's why you see a good match. But otherwise, there will be mismatches or gaps. For example, if we can find some gaps here, yeah, you see here A and G. That means this sequence was not 100% identical. You see here is an A, but here is an C. So this vertical lines actually tells you how well your sequence has matched. If it's exactly same nucleotide, it will be given as a dash, and if there is a gap, it will be shown as a dash in the sequence itself. But I don't see any dash here. But anyway, that's the basic thing. Now, there is one more thing that you can see is the strand plus plus. That means the directionality of the sequence. So plus, the first plus means where you how the database has this sequence, and the next plus is how your sequence has been aligned. So in this case, both of them have the same directions, like they are aligned in the same direction. Sometimes you will find this as minus. So in this case, your sequence has been reversed, and the nuclear in the sequence in the database has been the plus. So it will be something like this. So those are a few of the things that you can work around, play with it, and see how things go on. Right. So a step further, suppose after this you also want to know like how this sequences are actually aligned with each other, or how far they are from each other. Then you can also go for distance tree. If you click distance tree right away, as I did, you will get the entire set. If you want to select few of the data and not everything, then okay, let's not select everything and let's just do few of it. These are all homo sapiens. Okay, so if they're homo sapiens, more or less they'll be on the same branch. But yeah, you can select a few of these and then do the distance tree. It will give you only for those sequences, right? Oh, I just overlapped the previous one, but anyway. So as you can see, this is called a phylogenetic tree, right? These are the edge, these distance are known as the edges, and these points where it diverges are called nodes, right? So for better visualization, you can also do it in taxonomic name, but in this case it will be all homo sapiens, right? Because yeah, we just got a homo sapien here because we are using some variant of that particular gene. But anyway, when you are performing experiments, you can exclude the homo sapiens and you can select the other species with which it matches the best, right? So yeah, that's pretty much it about the phylogenetic tree BLAST. Now let me know if you have some question. or we can directly go to the next one. In the interest of time, let's just move on with the next one. So let's discuss about UniProt. UniProt is same as NCBI BLAST, but it contains mostly about the protein. So here it is like the universal protein resource. It tells you what the protein does, where it lives, what it interacts with, why it matters, things like that. Then there are mainly two flavors. One is SwissProt, which is the curated one, the manually curated, polished, the trustworthy proteins actually. And then we have Tremble or the translated. These are more about the predicted structures. These are not manually curated one, right? So what do we get from UniProt? We get the functions, the domains, the structure, pathways, variants, post-transcriptional modification, translation modification, sorry. And then what you do, you just, the same way, you just go to the search bar, you give the name of that, a particular protein, and you get what that protein actually does, right? Yes, then it also, the main part of this UniProt is here you can find the sequences, but basically it is a collection of databases. Like it will tell you where this particular protein is related to. You can also find links to the KEGG database, which are a pathway database, then a string database. So these are other databases to which it is linked to. So that's the most unique thing about UniProt, right? So yeah, let's do another practical quickly on what this protein actually does. UniProt. So yeah, we can go to UniProt and start searching. So this is the landing page of UniProt, right? So here you can just give the name of the protein and you'll get the subsequent data that is related to it, right? So if we just put insulin here again, our favorite protein for now, so you will see this result page here. This golden color one is the EMBL, the curated one. Sometimes you'll also see blue colored icons, which are the Tremble, the predicted ones. Right, so it'll tell you the entry name, the protein name, which organism it belongs to, the length of the amino acid sequence, and stuff like that. Right. So if you go inside, you will, on the left you will see these bands where you will find the taxonomy, the name, the localization, the disease variants that we just discussed with NCBI, then a post-translational modification, expression, and stuff like that. Let's go to the naming. So you see the same taxonomic lineage you can find from this, right, about that particular protein that you're searching. Then you can also see the accession key. These are the accession key, like the roll numbers that we saw in NCBI BLAST is the same for UniProt as well, right? And then again, we have this HGNC symbol. You'll come across HGNC more often when you are working with a disease, as I already mentioned. This is a standard nomenclature for a given gene, right? So this is about the taxonomy and naming. Let's see the localization, how and where it is there. So it is secreted, where, and more information about it you can find here in this page. But yeah, let's get back to it. Then you can also go for the disease variants, right? For example, if this H in the position of 34 changes to D, this is the disease which comes in, right? You can also check out the publication which is mentioned here. And similarly, let's say if in the 48 position, if the F changes to S, it gives rise to this. You can find, I mean, all the information regarding these disease variants are here along with the publication that you can search for, right? And then of course we already know insulin is related to disease diabetes. So whenever there is some problem with it, like some mutation, for example, in 55, if there is a change from R to C, we will get a diabetes type 2, and so on. This is these are the conditions when a person is born with such a disorder. For example, these nucleotide changes, they get this permanent neonatal diabetes because their chromosome is actually somehow mutated, like they have this problem in their gene. That's why the insulin that they will produce will never work, and the person will be born with diabetes, and because of these conditions, these mutations. You can read more about it in these publications over here. right? Then also we have about the post translational modification, right? How after the translation the protein is modified, there are more information about it, right? You can check here like where the sequence is changed and stuff like that. And yeah, just it depends upon the question you are asking actually, that you have to look for the answers. And here it is, mostly you will find these answers in these kind of annotations. So moving forward to expression, although there is this expression database linked here, like expression atlas or gene expression database, this is the geo database that we were talking about. So basically it will give us how the this particular gene is expressed between, let's say, a diseased and a healthy person, and you can do some bioinformatics analysis, like some transcriptional and this is called bulk transcriptomics or single cell transcriptomics. Depends upon what kind of data you're trying to analyze. You'll get these kind of variations, and you'll understand how the data, how the gene is actually fluctuating. So those information is stored in geo or expression atlas or array express. This is also called array express. So these are databases that you can find for the expression analysis, right? Then we have some interaction studies, like where the gene is, where the protein is interacting with other proteins and stuff like that. Then we have the structure of it, like the three-dimensional structure. So if you want to know more about this structure, we have to go to RCSB. That is the next database that we have, right? Here we already have the link to the RCSB. This is the good thing about UniProt, sorry, that it contains the cross-linking platform for all the databases, right? You want to find something, it will give you the links to the other databases as well. For example, for the structure, it will give you RCSB. For pathways, it will give you the KEGG or Go databases, right? And so on and so forth. So that's why this UniProt is still very popular and high in demand. So let's see from here itself, we can go to RCSB, I hope. If not, we'll just go to RCSB and just see what is, yeah. So this is another database, which is RCSB, and it contains the PDB structure. It's basically for the three-dimensional structure that you want to play with, right? So for insulin, you'll have the structure like this, right? You can check more information about it, like the alpha helices, beta sheets, how they are, and stuff like that. Here we just have alpha helix. But suppose you want to work with this protein, do some molecular dynamic simulation or something like that, then you can download the file, right? Not the FASTA file here, but we'll need the PDB file, PDB format. So let's say we have the PDB format over here. I hope I downloaded the correct one, but let's see if I try to extract it here itself. Okay, good. So if I try to open it, I'm using Discovery Studio. So here you can see this structure of the insulin molecule, right? So like this, you can also select the different kinds of things. For example, in insulin, we all know there are two chains, alpha and beta. So if you want to go only visualize the alpha and do some simulation over here, you can do that. Or likewise, if you just want to do on the B, you can do that as well. Then suppose you want to know the individual amino acids. These are the individual amino acids. Let's say you just want to know where the valine and other, let's say isoleucine, valine, and glycine are. Then you can just select them and deselect the other ones, right? It will also tell you this particular amino acids contribution to the PDB structure. Here we can see it's just undefined area and the alpha helix. That's why you see most of them are alpha helix, right? It's just alpha helix. But if you take a more complex protein structure, right, then you'll see beta sheets, alpha helixes, and other motifs. So those will be mentioned over here. So from here, you can select your amino acid bases that you want to work on and run your simulations. So simulations are nothing but you are providing a force field to it. Let's say this is just in a normal structure, right, in a crystal structure. But when they're inside our body, they have some force field around it because it's in the liquid, right? So basically we have to give a boundary of it in a solid structure, calling it that it's inside a solvent, and then it will work like that, how these molecules will be interacting with the surrounding molecules. And if you want to see the interaction of this one with another, let's say protein, then those force field will change, the bonds will change, right? So those things are done in the MD simulation, right? So yeah, that's pretty much it about the RCSB as well. So yep. Finally, I think we are in the last portion, which is the e-utilities that I wanted to check with you. So so far we saw about the databases that we can access with our browser, right? So but that is quite time consuming, right? So if you're working with thousands or millions of data and you want to do it fast, quickly, so how are you going to do it? So basically there are things like e-utilities, which is a command line tool. You can run it from your Unix environment, and you can get all the data there, do the automation, running batch pipelines, and it saves you a lot of time than manually scrolling through the pages over there. Right. So there are two main things. One is the e-search and e-fetch. E-fetch actually, e-search searches for the things. E-fetch fetches you the relevant data. For example, this is a e-search command that you can use. This e-search, you are searching for the database called nucleotide, and then let's say you're working with BRCA1 cancer. So you can just go over here and paste it, right? So basically eSearch DB is nucleotide, query is BRCA1. This is a cancer type, breast cancer, and then you want to limit your query till 5. So if you press enter, you'll get this kind of data, right? So it tells you the MCID, the nucleotide, how many counts and stuff like that. But this is not much useful, so let's see a more practical scenario. So let's say you want to get the gene bank format. I'll just show you the analog of it. So right, we were working with insulin, right? So let's go back to our insulin molecule. Maybe we'll just use the same insulin molecule here. Yeah, let's say I want to work with this accession, right? Let's say when you click on an accession, it will give you this GenBank page that we already saw, right? This thing. So what I want to do is, I don't have a browser. Let's say I'm just working on a terminal. I don't have a mouse. I just want to work with some Unix environment. Now I want to know more about that accession ID. I can just copy and paste it over here. The page you saw over there is exactly the same that is displayed over here, right? The locus, right? The places, the definition that we saw, the journal, the title. This is exactly the same. It's just parsing that inside your terminal now, instead of using a browser. So this is very fast. Instead of using mouse and clicking your way through, you can just get the data like this, right? And suppose instead of all the information here, you just want the faster sequence, let's say, that we saved. You can also do it like this. It will just give you the faster, the same thing that we did with downloading the file and all that. So instead of doing that, you can go for a quicker way. This is just for one entry. You can have multiple entries here. It will fetch all the data at once. Now for example, you also want to save this. You can do insulin.pasta, and it will save your file. So instead of going through that lengthier way, you can just use this method and see this file has been created, and inside this file you see something like this. All the sequences are here. So yep, I think that's that. We have discussed the GB, we have discussed FASTA, yes, and yeah, that's pretty much it with this tutorial. So yeah, thank you everyone for attending this meeting. I hope you were able to learn something, and at least you have some curiosity that you can work on any of the given disease that that interests you, and work further on it. So yeah, I'm Adhiraj. This is my email ID. You can send me any queries that you have. I'll try to respond it as soon as possible. And yeah, thank you to PhD Service Vibe for creating this free workshop. And yeah, thank you everyone.